Well, good afternoon, everyone. Welcome to today's webinar, Launching into the Next Phase of the NIH Brain Initiative. Thank you all for making the time to join us today, and thank you for your patience as we had a slight delay in getting started. I'm Janet Heischetter, Chair of the American Brain Coalition, as well as the Executive Director of the Dystonia Medical Research Foundation, a proud member of the ABC. The American Brain Coalition is pleased to host this webinar as a part of a series of educational programs offered to our members and affiliates. Today's webinar is a follow-up to a Congressional Neuroscience Caucus briefing held in February. ABC members requested regular updates on the Brain Initiative, and we are pleased to work with our esteemed speakers and moderator today to provide this update. We're all painfully aware that studying the brain is very challenging, and neurological disorders, and we include psychiatric disorders in this, are among the most disabling and costly chronic illnesses impacting some 50 million Americans. The Brain Research through Advancing Innovative Neurotechnologies Initiative, or the Brain Initiative, is advancing science, discovery, and the development of tools that are proving to be instrumental in accelerating progress toward understanding the mysteries of the human brain. The Brain Initiative has also devoted funding to studies that will have direct impact on patient care. Today, we're honored to have as our speakers Dr. Walter Koroshetz, Director of the National Institute of Neurological Disorders and Stroke, and Dr. Joshua Gordon, Director of the National Institute of Mental Health. We're also pleased to welcome our moderator for the Q&A session, Dr. Amy Adams, Director of the Office of Scientific Liaison at NINDS. The National Institutes of Health is both a partner and a leader in this important initiative. 10 institutes and centers work with numerous government agencies, private partner foundations, institutes, and companies in order to successfully accomplish the Brain Initiative's mission. The NINDS and the NIMH are the leaders in this initiative. We're grateful to Dr. Koroshetz and Dr. Gordon for their work on the Brain Initiative, their update and remarks today, and for the work that they do every day in support of this community. Before I turn it over to them for their reports and their presentation, just a quick operational note. We welcome your questions. As a participant, you are in a listen-only mode, but you can submit your questions throughout the presentation by utilizing the chat box function on your screen. Also, it's important to note that this webinar is being recorded and it will be available on the ABC's website. It's now my pleasure to turn the program over to Drs. Koroshetz and Gordon.
Well, thank you very much, Janet. It's a pleasure to talk to folks today about what I think is an incredibly exciting program that's being run under the name, the Brain Initiative. And this is a program that's been funded by the National Institutes of Health, but also other federal agencies, particularly National Science Foundation and the Defense DARPA program and the intelligence agency's IARPA program. And as we'll talk about, the N in Brain Initiative stands for Neurotechnologies, and the program has had some really exciting advances over the last couple of years that I think are really going to change how we study the brain and also how we treat patients. So as Janet mentioned, the problems that we're dealing with are those of trying to help people who are suffering from neuro and psychiatric disorders, and I'd also include substance abuse disorders as one of those. So we often go by the name NMS disorders, neurologic, mental health, and substance abuse disorders. And these are the leading cause of disability in the United States. And so you can see here on this graph how they stack up against many of the other very common conditions. And DALYs are disability adjusted life years. So this is a disability over the life of an individual, trying to quantify that. And just to add, those of you who are reading the newspapers know that this country is in a really terrible crisis in terms of the addiction problem that has led to overdose deaths that are just now occurring at a rate that was higher than occurred at the peak of the AIDS epidemic. And if there ever was a brain circuit disorder, and we'll talk about brain circuits as the focus of the Brain Initiative, certainly opioid addiction is a classic. The brain is completely rewired by the exposure to opioids. So we know, and I'm sure many people on the phone know, just how difficult these disorders are. And so how do you actually make progress? People have tried for years to make progress, and we have, certainly in certain conditions, but never as fast as we want it to be. Well, the Brain Initiative is kind of focused around, or revolves around, this idea that new directions in science are launched by new tools much more often than by new concepts. The effect of a concept-driven revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that have yet to be explained. And that's a quote from Freeman Dyson, who is a physicist and philosopher at Harvard. And so the idea here is, can we develop the kind of tools that'll open up the brain, particularly how our circuits work, that will enable us to better understand how to treat people with the disorders we talked about. And truth of the matter is that certainly over my career, technology has expanded to the point where in 1974, we were very excited to see this very smushy picture of a brain of an individual on a CT scan. And really all you can see is the ventricles. But prior to that, there was no way to actually see in the brain of an individual before examining it after death. But look what happened in the ensuing years, and look at the kind of definition we now have with this new tool, which is really high field MRI scanning. And that completely changed how we practice neurology, which is oftentimes based on understanding the clinical and pathologic correlation. So there's something to see in the brain when you examine it. And so the imaging gave us the ability to try and see that pathology in living individuals. But, you know, the disorders we mentioned earlier, something like addiction or even chronic pain, there's no structural abnormality. And it's really a matter of altered circuit function, circuit activity. We really don't have the ability to see that type of information. Now, in terms of the Brain Initiative, one major goal of the Brain Initiative is to try and develop a method of seeing the connections between all the different brain areas. There are 85 billion neurons in the human brain, trillions of synapses and connections. And amazingly, there are technologies like you see here. This is called the clarity technique of being able to make the brain transparent, so that if you have stained the brain as you have here with, say, a neuron-specific fluorophore, then you can remove the brain, remove all the lipids, which is what blocks the light from going through the brain. And now you can see in all the exquisite detail all the different cells and their connections. So technologies like this, clarity, enable us now to actually think about the understanding brain connections, structural connections, in a way in which we could never do so before. There are now laboratories that have electron microscopes running, five of them at a time, 24 hours a day in, day out, to get the entire electron microscopy level connection, say in a Drosophila brain, and then potentially moving on to the mouse brain. So the technologies for understanding structure are moving very quickly, and these are one of the foci of the Brain Initiative. But in fact, the Brain Initiative is more ambitious than that. The Brain Initiative is trying to to actually go the next step and to be able to see activity as it moves across these structural connections in the brain. And these tools, we think, will build the foundation we need to reduce the burden of brain disorders. So what do I mean by that? I use this as an example. As a neurologist, we have, as I mentioned, examined the patients, made lists of the neurological signs and symptoms that the patient has, and then we've examined the brain to try and understand what the pathology was that gave rise to those symptoms. But in fact, the patient's symptoms and signs are due to dysfunctions in the circuits. We've never had the ability to actually see the circuit dysfunction. We can only infer it from the pathology, and there are certainly lots of disconnects over time that we know about but try to hide under the rug. One example is the finding that there are, in aged individuals, one can look in the brain and see fairly dramatic changes of what looks like Alzheimer's disease, and yet the person is functioning at a really high level. So the person doesn't really care about how much amyloid or how much tau, but how it affects their memory circuits or other circuits in the brain. So trying to understand how to see those kind of disordered circuit activity is the goal of the Brain Initiative, the eventual goal of the Brain Initiative, to see the circuits in action, understand how the brain controls our ability to move, plan, execute, remember, and then how to monitor or modulate circuits to improve function. So in neurology, we have the example of deep brain stimulation for Parkinson's disease, really dramatic changes that can occur in a patient's symptoms when the brain is stimulated in a particular area, at a particular frequency. And it's really quite crude technology that's been successful there. But we haven't had those same successes in many of the other areas where there's brain circuit disorders such as depression or schizophrenia, bipolar disease, obsessive compulsive disorder, chronic pain. And so we think that there's a whole new way of helping people by learning how to monitor and modulate circuits for improved function. And then to understand how these circuits go awry to cause neuro, mental, and substance abuse disorders, so that we can then either change that to either normalize the circuit activity or develop compensatory circuit activity that improves patients' function. So that's the overall final goal of the Brain Initiative from the clinical standpoint. But there's a great deal to be learned about the brain, and we're doing that as well as at the same time trying to improve our therapeutic abilities to monitor and modulate brain activity. So I think that I'd like to talk a little bit now in depth about the Brain Initiative and the different parts of it. They are all focused, as I mentioned, on understanding brain circuits. And so this is only a piece of neuroscience, but a piece that has really been difficult for people to get their hands around. And we'll talk about what some of those barriers were and how the technologies are breaking through those barriers. But let me break it down for you in terms of these color-coded areas here. The first in red we call discovering diversity, which is really understanding what are all the components of the brain. So these are the cells, but there's different types of cells, and they have different properties, and they have different functions and circuits. And so to understand those circuits, we really need a parts list of all those different cell types. And next, as we mentioned already, which is trying to understand the connections. So these are developing maps at multiple scales meaning the connections between neurons that are occurring, at the nanometer or micron level, or between neurons and glia, or glia and the vasculature. So these developing these maps at these small scales, but then trying to understand how not only do cells adjacently interact, but then how they interact in a nucleus, how that nucleus interacts with widespread brain regions to eventually end up in really what can be very complex behaviors. These multiple scales is probably the biggest challenge for the Brain Initiative, because we're going from the micron to the many centimeter level, and we're dealing with really trillions of connections. So that is probably something we're not going to be able to figure out in the next 10 years, but we can develop the technologies that future generations can fill in as we go along. As I mentioned, the idea is to get technologies that can see circuit activity in action. So not just the static picture, but how brain activity gives rise to behavior, how brain activity changes during development, changes with exposure, practice, for instance, or stimulation. And what we'd like to do is to go beyond just developing correlations between, say, a pattern of cell firing and the behavior, but actually demonstrate causality, that firing pattern actually does give rise to that behavior. And we can do that now if we can get enough data on how the neurocircuit is behaving, develop a model that links the neurocircuit activity and its patterns of activity to the behavior, and then actually intervene and change the neural firing pattern and predict what that will do to behavior in an animal, for instance. That gets us past just correlation, but gets us to really the link between the causal nature of the neural activity and the behavior. This activity that we're talking about gets us into a space that we've never been before, because we've never been able to see network activity in order to develop and understand the rules that the brain is using for information processing. So in the past, most of our work has been done by recording from 1 or 2 neurons at a time. But as you'll see, and as you probably have guessed, that in any activity, there are tens of thousands of neurons that are taking part in that activity. And the rules that govern how that pattern gives rise to behavior have never really been explored before, because we've never been able to get that kind of rich data. So we think that by collecting the data and doing these analyses and correlation and causal experiments with behavior, that we'll get to underlying fundamental principles of how the brain works that really eluded us so far. And the final goal is to move from the experimental conditions that are best suited to the technologies and kind of move those along to help us understand how human brain networks are working, to give us the unique types of behavior that only humans have. And again, as we mentioned, to understand then how these circuits go awry in disorders and how we can modulate those circuits for health. So that's kind of the big picture of the Brain Initiative in all its pieces. And we have, I think, been very successful in moving each of these pieces along, although I must say that as all things in science, certain things move faster than others. So in this slide, you can see where we are now. The Brain Initiative funding for 2018 is almost $400 million a year. In 2017, there were the funding was about $260 million. There were 110 awards, and they span all these color-coded areas that we talked about in the previous slides. We've been able to engage hundreds of investigators, and these investigators come from all over the US and also from different countries working with US investigators. So this has really grown into quite a remarkable project. And we're really looking forward to the ability to move the project forward because we have been the beneficiaries of congressional interest in this program. So that here you can see what's happened to the budgets over time since the BRAIN Initiative was launched in 2013 and funding started in 2014. It's ramped up to, as I mentioned, a projection of $400 million in 2018. But the 21st Century Cures bill also provides funding in green in these bars going out to 2026. That is on top of the increases that came to the base budget of the NIH for this project. So we have really quite remarkable enthusiasm on the part of the Congress in affording us these resources to carry out the project that I described. Oops, just go back one. Got a little quick there with the slides. Here we go. Okay, so just to recap, the BRAIN Initiative was announced in April of 2013. What I hadn't mentioned, it should have, is that the progress and the roadmap that we've been following came out of a report called the BRAIN 2025 report that was released in June of 2014. That's available on the web. You could just Google BRAIN 2025 NIH. And this is a really scholarly and wise guideline to the NIH for carrying out the BRAIN Initiative. We've really stuck very closely to this over time. The BRAIN Initiative is managed by multiple institutes, 10 institutes at NIH. The leads are mental health and NINDS. We have a BRAIN multi-council working group that looks at all of our activities as we go forward, both those projected activities and the activities underway, and provides feedback to the NIH. We have annual meetings, which are quite exciting meetings. The last one brought in over 1,000 different folks to learn about all the different parts of the BRAIN Initiative. There are BRAIN initiatives that have been stimulated across the world. The European BRAIN Initiative has launched actually almost consonant with ours and is focusing on particularly technologies that can be built off of knowledge of brain, such as neuromorphic computing, for instance, better robotics, and there are BRAIN initiatives in Korea and China, Japan, and we have a BRAIN Initiative Alliance, which brings together all the partners in the U.S., which includes private groups such as the Simons Foundation and the Kavli Foundation, in addition to the other federal partners, particularly National Science Foundation and DARPA and IARPA, as I mentioned. So we are really quite lucky and happy about how things are moving so far. And in fact, there have been a tremendous number of publications that have come out for the BRAIN Initiative about tremendously new and powerful technologies to look at brain circuit activity. And we'll talk about some of these as we move ahead. Now, I guess one of the areas in the BRAIN Initiative that has exploded the highest, I guess, at this point, not to take a riff off of the Hawaii volcano, but the ability to actually understand all the different cells in the brain. As I mentioned, there's 85 billion cells in the human brain, 8.5 billion in the mouse brain. How could one ever figure out how we were going to get a parts list of all these different cell types? And in fact, the technologies really came through on this. Josh, do you want to talk a little bit about this? Because this is certainly been a project that the National Institute of Mental Health has been the lead on, and really quite exciting.
So we knew from the early days of the Brain Initiative that we were, in order to be able to measure activity in specific cell types and manipulate those cell types and drive behavior and learn about behavior, that we would have to understand what the different cell types in the nervous system were. And that was a very daunting task 5 years ago. But some really amazing technologies have emerged, so, particularly around single cell transcriptomics and other omic technologies that really have facilitated high throughput interrogation of cell types. So building on the strength of those technologies, this past year we began funding a $250 million effort over the next 5 years. The prime focus of that effort is to map the number of cell types in the adult mouse brain, as well as begin some pilot projects to scope what it would take to do the same thing in human and non-human primate brains. These are collectively known as the Brain Initiative Cell Census Network. The network has 9 funded projects that support a brain cell data center that will coordinate data acquisition and create an online atlas, as well as a collaboratory that and the cell census centers that will actually do the work of cataloging the various cell types. And then the fourth component is really a collaboratory built around targeting these technologies for use in humans and non-human primates. So there you go. The technologies are really wonderful and sophisticated in that they allow us to identify individual cell types, such as shown in the video on the left. Is it going to click the one on the left? And target those with markers that we can follow the anatomy of these cell types. And then on the right, of course, images showing how we can image the activity in these various cell types. The idea will be that once the library, if you will, of cell types is established, and once we have a true cell type atlas, we can apply these technologies to characterize those cells anatomically and physiologically, as well as understand their relationship to behavior.
Yeah, and so I guess the other point to make is that with these new optical techniques where self-firing lights up the cell, scientists can get data on thousands of cells simultaneously, which brings to the forefront how do you analyze all this data, and that's also going to be a major new opportunity for the future that we've never really had the ability to see all these large number of cells interacting in real time.
I'm pressing the wrong button here, I guess. Sorry, folks. Here we go. Okay. So we have big plans for this atlas. The idea is that we'll get essential knowledge on hopefully thousands of brain cell types, but also their 3D organization. Where in the brain do they sit? And this, in the end, will result in an open access 3D digital mouse brain cell reference atlas, as well as, once the anatomy is in hand, a comprehensive neural circuit diagram of the mouse brain. And we think that these will be incredibly useful precision tools that will facilitate individual investigators' studies on how to monitor, map, or modulate the activity of those specific cells in relationship to behavior. The fact that we have begun the cell census doesn't change the fact that we are still looking for and funding efforts to develop new technologies for circuit mapping, as well as interventional tools for functional circuit interrogation across scales. On the right-hand side here is an innovative microscope, a really dual microscope, developed by Michael Crair at Yale University, that can produce the kinds of images you saw from a similar scope developed at Janelia Farm, where you can monitor the activity of a large scale across the entire brain at the same time that you're monitoring it in very fine detail in specific brain regions at a cellular level. And then on the left, we now have the technology to be able to image the entirety of certain smaller brains, such as the insect brain, demonstrated here. Anything else you want to say about that one?
No, that's good. And the next slide is another image from Michael Kerr's lab. And so this is a awake behaving animal where the microscope is engaged, where the animal is behaving with a particular task. And what you see is this cortex-wide activity. So with this task, huge expanses of the brain are activated, and again it opens up the ability to see networks like we've never seen before. But then on the right-hand side, one can zoom in and actually get single cell activity that is making up this whole brain activity. And this is the kind of technology that's now available to bring just tremendous amount of data in. How to make sense of the data becomes, again, the next challenge.
So we can image large scale across the entire mouse brain and circuit scale within certain areas of it. But these same technologies allow us to develop wiring diagrams for whole organisms like zebrafish here on the left, as well as to monitor the activity for the of the individual neurons within the entire zebrafish brain. On the right-hand side, we move from calcium imaging and using fluorescence activity to image activity of the brain to novel technologies to look electrophysiologically at the activity of neurons. And that has some advantages in terms of temporal resolution, but the methods to do so to maintain quality recordings over time have been challenging. In particular, with indwelling metallic electrodes, one tends to get immune reactions that can weaken neural signals that you record over time. And of course, if we want to use these devices for things like brain-machine interfaces for therapeutic reasons, we need them to last a long time. So brain investigators, including Cynthia Chestak here, are developing invasive probes, in this case, that record electrical activity, but that, as you can see from these images of greenly fluorescent neurons here, evoke very little, if any, immune reaction, so that the signals from those neurons can be recorded over long periods of time without degradation.
Right.
Finally, we need not just to measure, but also manipulate. And ideally, we would measure and manipulate all at the same time. But these probes that we use to do this measuring and manipulation can be quite bulky and challenging to design. And so Paulina Anakiva at MIT, among others, are working on ways to make probes, that simultaneously allow us to image and intervene, but all combined in one package. In this case, using a technique such as that used to draw out optical fibers. But these techniques allow inclusion of not just optical fibers to bring in light or push light out of the brain, but also channels for introduction of chemicals, as well as electrically conducting channels as well, so that one can imagine doing optogenetics to influence brain activity. One can imagine doing calcium imaging to measure brain activity. At the same time, you're measuring electrical activity and introducing pharmacological agents into that part of the brain. These tools are going to be extremely useful for investigators to examine the brain from multiple perspectives simultaneously.
And so we're going to shift now a little bit to talk about the brain initiative activities that are going on in humans. So human imaging and modulation is where we'd like to go. And we are certainly limited by the physics of the brain and the skull in doing this safely. But we do have some technologies, as seen here on the lower left, the resting state MRI scan. And there are instances in which, as you can see here on the right, that there are clinical indications for recording and stimulating brain in patients, for instance, with Parkinson's disease. And so we are working at this space to improve the technologies that can be used in patients. So we have programs going on to develop new imaging technologies, maybe one to hopefully someday replace MR, trying to understand the biological basis of the type of signals we can get from MR or EEG or MEG, for instance, so we can make better predictions of what's going on in the brain from these signals that are obtainable from outside the head. And also ways of non-invasively modulating brain activity, either through electrical activity or some really quite unique genetic techniques that we'll talk about in a second. What has changed most recently is the fact that there are now devices that are used in deep brain stimulation, say for Parkinson's, in which one can not only stimulate the brain for clinical benefit, but also record electrical activity from the same electrodes. And this has allowed investigators just in the recent years to collect a tremendous amount of data from the inside of the brain in patients while they're trying to treat the disorder and learn how activity can be used to regulate the devices, develop what we call closed loop devices, where the stimulation is really linked to the activity that you're trying to modulate. And the next slide is an example of that. In the case of essential tremor, the patient has a severe shaking that impairs their ability to perform fine motor activity when they actually move. But at rest, they don't have any tremor. And so the question is, as you can see here in the video, is there a way to stimulate the brain just when it needs that stimulation when the person makes an attempt to move? So now with one of these recording electrodes, you can see in the power channel and detection channel that activity occurs when the patient is actually making the intent to move before they even move, and that is what turns on the stimulator. So the brain is only stimulated when the patient actually needs it. So of 130 of the 345 brain awards that we had in 2017, there were 80 projects involved human subjects research. And these are targeting 9 distinct disorders. Many of them have to do with what I mentioned, new methods for deep brain stimulation. But in conditions other than Parkinson's, depression, obsessive compulsive disorder, tremor, as I mentioned. And we also have a project trying to stimulate the brain in people after stroke to improve recovery after stroke. There are also a number of investigators funded by the Brain Initiative that are studying patients who have electrodes over the surface of the brain because they're mapping out where their seizures are coming from to inform epilepsy surgery, but also gives an opportunity if the patients are willing to participate in research to understand the neural activity that undergoes things like language or speech function. So we have quite a bit of very rich investigations going on in the human condition as well. One area that we're really interested in getting work going on is, as I mentioned, in the area of pain and addiction. And these are just some recent graphs to show you the tremendous increase in deaths that's occurring in the US due to addiction. And much of this started with the use of opioid medications for pain. And what we'd like to really try to do is to understand the pain system to a point where we can modulate the circuit activity in ways that don't require opioids. So the NIH has been pushing for an all-out campaign, a research campaign, to try to develop new ways of not only reversing overdose, preventing death, but also treating addiction and developing non-addictive interventions for pain, especially chronic pain. As one might have guessed from the discussion so far, that the Brain Initiative, in its movement towards understanding how to modulate and monitor neural circuit activity, particularly in humans, brings up ethical issues that need to be looked at carefully and integrated in the research as we move along. And so right from the beginning, the Brain Multi-Council Working Group, which informs the NIH on its progress, developed a division that's entirely devoted to integrating neuroethics into the research. And we feel, quite confident that these medical issues that come up as we develop these new technologies can be advanced with the strongest ethical background. So this group is working to develop guiding principles of neuroethics in the Brain Initiative. It's an ongoing dialogue between neuroscience researchers, neuroethicists, and other stakeholders. And we think that it's an ethical imperative to actually pursue neuroscience research in this space to make a difference for people with mental, and substance abuse disorders, and that the neuroethics is really vital to the neuroscience research. We are not, again, the NIH is as I mentioned, not alone in the Brain Initiative, and we have a Brain Initiative Alliance, which has a website, which I'd encourage folks to look at and tell their friends to look at, but it's a platform to kind of communicate all the type of activities that are ongoing in the Brain Initiative, not just from the NIH, but from the other federal partners, particularly the National Science Foundation, IARPA, and then the private groups, the Allen Brain Institute, the Ph.D. and the Simons Foundation, and the Kavli Foundation.
So we have also every year now for the 4 years running a meeting of all the investigators who are funded through the Brain Initiative. We're pleased to report back on our April 9th to the 11th, 2018 meeting, which subjectively was the most dynamic and exciting we've had, with a lot of people really jazzed about the technologies that are coming out and the findings that those technologies allow us to make. There were 5 major plenary sessions, all archived on NIH Videocast. If you'd like to take a look, you can find them. We had 36 research highlight talks, 3 different poster sessions with over 400 posters, and we gave out this year 32 trainee travel awardees. We recognize that training is an important component of the Brain Initiative. There were lots of panel discussions, agency-specific focus sessions, over 1,200 registrants, and the meeting continues to grow as the Brain Initiative grows. Early feedback suggests again that it was a really dynamic and exciting meeting for those who were able to participate. Want to point out that in addition to the Brain Alliance website, the Brain Initiative has a website that has listed all the brain funding opportunities. So those are listening in or interested in trying to figure out what we have available right now for you to apply in. They're there on the website. It also lists all the Brain Awards sorted by research topic and investigator, so you can get a sense of the kind of work that we've been supporting. And finally, we recognize that as we approach the midpoint of the Brain Initiative, which will be in 2020, that we can't assume that the plans we laid out when the Brain Initiative was launched are necessarily the right ones to follow through the second half of the initiative. The Brain 2025 report, which was drafted by an independent group of scientists working under the auspices of the advisory committee to the director of the NIH, is really a wonderful and comprehensive document that points out the work needing to be done in these various areas that we've been discussing today. But we recognize that, especially in some areas where the technology development has really taken off, we might want to review these goals. to review the progress that we've made and figure out what new goals we can now accomplish in the same time frame because of technological advancement. We also want to take a pause and see whether there are any areas that we still need to focus on technology development because the technologies aren't where we need them to be in the long run. And so we've asked a new panel of outside scientific experts to take this on. They're chaired by Catherine Dulac and John Monsell, and they include members from really across the scientific spectrum with diversity on a number of fronts, and also include ex officio members from our various federal partners. So this group has been tasked with having, with looking over the 2025 report, figuring out what needs to be changed. And in that endeavor, they're going to need help from all of you. They're going to be having lots of opportunities for public input, including, but not limited to, requests for information and public events across the country to get feedback on as they go about revising Brain 2025. And if you don't want to wait, you can give us feedback now. brainfeedback@nih.gov is the general address, or you can contact Walter or myself directly, and we're happy to forward it on to brainfeedback@nih.gov. Or if we have the wherewithal to address it ourselves, we'll do so. So thanks very much for your attention. And
well, thank you very much, Dr. Gordon and Dr. Koroshetz, for that wonderful overview and update. I think I can safely say on behalf of patient organizations, this is way cool and very exciting. So we're really appreciative for this report. At this point, I'll turn it over to Dr. Adams, who's going to moderate the questions that we've received. Dr. Adams?
Thank you so much. Yes, and I'd like to encourage everybody logged on to please send in questions, and everyone here is really excited to engage with you and answer them. One of the first questions that we've received was someone who wrote in and asked what sorts of activities are funded under infrastructure for brain?
Well, I think I guess the one that is probably the most important is developing a data infrastructure for all the different components of the Brain Initiative, because as we've told, as you've seen in multiple different slides, the amount of data that's coming in is really astronomical, and it's of high quality, and we would like it to be available to investigators around the world. So we are building data infrastructures along with the different projects that we think will really change how neuroscience is done in the future, because the type of data and the analysis that's needed really outstrips what we have ever done before in terms of the needs for mathematicians and computer scientists to really be able to dig into this data.
I would add that one of the things that we're asking the 2025 report, the version 2.0 team to look at is how we can improve on dissemination, and that may indeed involve establishing some infrastructure programs. But we'll see as we approach the halfway point how much we can do in that area.
That's great. That leads directly into the next question, actually, which is when do you anticipate having the updated Brain 2.0 report ready?
June of next year.
Great.
June of 2019.
Given the broad scope of the Brain Initiative in general, how do you think you'll know when the Brain Initiative is done? It's not exactly like the Human Genome Project. And maybe even that, it took a while for that to be really done.
Well, I just want to emphasize, I think I mentioned it earlier, that the Brain Initiative is one part of the neuroscience that's funded out of NIH, and it's focused on circuits particularly. And it doesn't really end until we have a comprehensive knowledge of how the brain works. So I would say this is a stepping stone to the future. I think it's a big step, but and I think that there will be deliverables that will impact patients, in the near term and also in the long term. So I think it's an important program. It does have an end date on it, but I think that the science that it spurs will go on for many generations.
Yeah, I would add, when will we know it's done? Well, we'll know it's done when 2026 rolls around and the Brain Initiative allocation runs out, and it will be up to Congress to decide whether they want to have a Brain Initiative, a second version of it, or whether they have other priorities they're interested in. But the good news is that what we hope to accomplish with the Brain Initiative is establish a set of tools that each of the neuroscience initiatives, each of the neuroscience institutes, can have their investigators used to make fast and strong progress against towards knowledge that will help those suffering from the illnesses that the various institutes are responsible for. There will be some, I think, products that we can look back on and say the Brain Initiative did, akin to that first human genome sequence, if you will. I think the mouse brain cell atlas will be one of those products, and with a little bit of luck and some ingenuity on someone out there listening to this, we can anticipate non-human primate and brain atlases as well. There will be new methods of imaging and of measuring and manipulating activity that will be in widespread use that we can point to as products of the Brain Initiative. But it is my hope that the Brain Initiative doesn't stop until everyone with a brain disease is cured. And that means that the Brain Initiative, from the larger perspective, is going to continue as a collaborative effort of all the brain institutes, even if it doesn't have the specific moniker, and even if it doesn't have the specific allocation.
Great. So there's another question that's come in talking about coordination, both with the other federal research agencies and with the international and European brain efforts. How is that coordination taking place, and is that helping to spur progress?
Yeah, so we have been really lucky at NIH to have teams of program directors who have been working on all the different color-coded areas of the Brain Initiative that you saw. And so this has been the Army that's coordinated the effort at NIH. But in addition, there have been similar type of interactions with the program directors and the senior level folks at the other federal agencies of the U.S. And I would emphasize the National Science Foundation has just been an incredibly strong partner and very complementary to what the NIH does in terms of the Brain Initiative. And so I think that has been a real pleasure to work with other agencies. And in terms of the international efforts, again, these are coordinated generally at the level of the senior staff communicating what they're doing and sharing their plans, but also getting the investigators together. So at the Brain PI meeting, it's not just the NIH investigators, but also the National Science Foundation brain investigators and investigators from the European Brain Project or from other countries coming together and sharing.
Okay, great. I think that pretty much concludes the time that we have available for questions.
Thank you. On behalf of the American Brain Coalition, I just want to thank again Dr. Koroshetz and Dr. Gordon for their presentation and for sharing this information and for the work that you're doing every day in support of the Brain Initiative and also just all of the other things that you have in support of this community. Also, thank you, Dr. Adams, for moderating the great questions that our participants generated today. Thank you again to everyone for participating, for making the time. And again, just as a reminder, this webinar has been recorded and will be on the American Brain Coalition's website. So thank you, everyone. We wish you a good afternoon.